Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to perform differential binding analysis in ATAC-seq data. Previously, I showed you how to perform this analysis in ChIPseeker data. Remember the major problem we face in ChIPseeker data? The peaks identified in each sample are different. So we have to create a consensus peak set, quantify this peak set across all the samples, and then perform differential analysis and all other downstream analysis. You can still use this method for ATAC-seq data analysis. However, this method involves in multiple tools along the process. So in this tutorial, I'm going to show you a new way that integrates all the analysis in one package, from differential analysis to annotation to enrichment analysis and then to visualization. The new method relies on one R package, DiffBind. Compared to our old method, the pros of DiffBind include specialized for chromatin data, integrated pipeline, statistical flexibility, and built-in visualization. The major disadvantage of DiffBind is probably its black box nature. However, as a biologist, you probably wouldn't care about what's going on in the code. DiffBind relies on two other R packages, DSeq2 and HR. DSeq2 generally provides more conservative results. HR may identify more differential sites, so pick the suitable one for your study. We'll perform the analysis in R, so on your laptop, open your RStudio, install all the essential packages. Here is DiffBind, and all other three are genome annotation files. We have mouse data, so we install mouse genome annotation files. If you have human data, you can install human annotation file. Data table for data manipulation, and load all the packages you just installed. The input of DiffBind is a sample sheet with this structure. It has a sample ID column, condition or treatment column, BAM race column which shows where the BAM files are on your computer, and peaks column which shows where the peak files are on your computer, and the peak color column which shows the types of peaks. You can make this sample sheet in Microsoft Excel. Just make sure to save it as a CSV file. Or you can make the sample sheet in R. Just put the contents of each column into a vector, and then assemble the vectors into a data frame, and then save the data frame onto your computer. Note the peaks here are called by max2. Yes, the max2 output can be directly used as input of DiffBind. Now let's read in our data using DBA function. The input is the sample sheet we just created. The output is the DBA object. Here's a summary of the DBA object. We have the sample information, the number of reads in each sample, and the fraction of reads in peaks. Next, we create a consensus peak set and quantify the peak set across all the samples using the dba.count function. Input is our DBA object. Summits, that means we center all the peaks on the summits, extend to the flanking region of 250 BPs. We merge the peaks overlapped with 2 BPs and quantify the merged peaks. You can, but you don't have to, extract the count matrix using the dba.peakset function. You can visualize the sample correlation in a heat map or a PCA plot. To make these plots, just call the built-in function plot heatmap or plot PCA. The input is just the DBA object from our previous step. You can also specify which label to show in the PCA plot. For differential binding analysis, we first establish our contrast. Input is our DBA object. Categories, our condition. We need at least 2 samples in each group. We call dba.analyze to perform the differential binding analysis. You can visualize the summary of the results, or you can extract the differential peaks using dba.report. Input is our DBA object. We can use FDR less than 0.05 as our threshold. convert the GRange object into a data frame, and you can save the table to our computer. The table is exactly the same as our DG table in RNA-seq data analysis, except we have peaks instead of genes. To associate differential peaks with genes, we can use annotated peak function. Input is our differential peak table. We need genome annotation files for mouse, TSS region, 3 KB flanking regions. We can convert the results into a data frame and save it onto our computer. This is the format of the resulted table. We have peaks, distance, and genes. DiffBind provides very handy built-in functions for visualizing differential peaks or samples. You can generate these plots with one line of code. For example, plot Heatmap, plot PCA, plot MA, plot Volcano, plot Pi, etc. You can also specify the format of the pictures. For example, PNG, JPEG, or PDF. You can specify the name of the plots, the dimension, and the resolution. This is all I have for today's tutorial. Subscribe if you like it, and I will see you in the next video.